All right, so here we go again. Okay, so in meiosis, one of the things that will be different is that here there's going to be 2 rounds of cell division. So instead of you seeing prophase, metaphase, anaphase, telophase, you're going to see prophase 1, metaphase 1, anaphase 1, telophase 1, and then we start it all over again. So then it becomes prophase 2, metaphase 2, anaphase 2, and telophase 2. So the first round and then a second round. As a result of those 2 rounds of cell division, then you'll end up with 4 cells as opposed to the 2 cells that we saw in mitosis. Here, meiosis produces 4 cells, and all 4 cells are going to be different from each other, and they all have half the number of the chromosomes. So here's a chromosome number 1, a number 2, a number 3. A number 1, a number 2, a number 3. So they're no longer in homologous pairs, and that's what's going to make this a haploid cell, because its homolog is in the other cell over here. See, here's mom's number 1 in red, here's dad's number 1. So see how the homologous pairs are now in separate cells? You only have one member of a homologous pair. There's the haploid. So again, this is still this idea of this homologous pairs. So we'll have 2 rounds of cell division, and so again, it's meiosis 1 and meiosis 2. So prophase 1, metaphase 1, and then the second round. Then they call it 2. Okay, so some of the things that are going to be really different in this process is that in meiosis, notice when we get to metaphase, so here's metaphase 1 of meiosis. So here's one of the major differences that happens in meiosis that we didn't see in mitosis, and that is the homologous chromosomes actually find each other, and then they line up side by side, side by side, side by side. In mitosis, they all just lined up right down the middle, not side by side, just one on top of the other. And the homologous pairs, they didn't even care about each other. It was just get to the middle of the cell. And here, they actually have to find each other. And when they find each other and they do this, it's called a tetrad. They form a tetrad because tetra means 4. It's a scientific Latin for 4. And so they form a tetrad. So here's another tetrad. Homologous pair number 2 here lined up side by side in the middle of the cell. So this is metaphase 1. So in the first round in metaphase, the homologous pairs are side by side. Then in anaphase 1, what gets pulled apart are the homologous pairs. Compare that to mitosis. So I put that worksheet in there for you to do a comparison of mitosis versus meiosis. And these are the critical steps in there that you really want to pay attention to, because in meiosis, in metaphase 1, the homologous pairs are side by side. Compare that to how you draw metaphase in mitosis. Okay, and then check out your differences when you're looking at anaphase. So this is anaphase 1 in meiosis, and here the homologous pairs separate from each other. Then when cytokinesis occurs, you now have already created your haploid cells. So in the first round of meiosis, the haploid cells are produced, because haploid means you have both members of a homologous pair in your cell. And right here, they do not. See, this is number 1 from your mom. Here's number 1 from your dad. Yes, they are sister chromatids and they've replicated themselves, but these two make up a homologous pair. and they are now in two separate cells. So right now, you can say that this cell is haploid and this cell is haploid. So in the first round of meiosis, some really significant stuff takes place, and that is the lining up side by side of the homologous pairs, the separation of the homologous pairs, and the production of two haploid cells. Those two haploid cells are now going to go through meiosis 2, the second round. And so in the second round, both cells are going to be going through it simultaneously. So these are those two cells right here that were produced, carried down to here. And so now in the second round of meiosis, it looks just like mitosis. So here's metaphase 2. Chromosomes line up right down the center of the cell. Looks just like metaphase of mitosis. So compare this to your mitosis drawing, and you'll say, wow, that looks just like mitosis. Then in anaphase 2, the sister chromatids are pulled apart to opposite sides of the cell, and then in telophase 2, the cell splits down the middle. Cytokinesis, the cytoplasm is separated, and here we've got 4 haploid different cells. Different because this one here has chromosome number 1 from my mom, and this one here has chromosome number 1 from my dad. And if chromosome number 1 has hair color on it, and I got blonde from my mom, and I got black from my dad, then if this is the egg that I ovulate this month and fertilization takes place, the offspring has information for blonde hair. If this is the egg that I ovulate this month and fertilization takes place, the offspring has information, my dad's information for black hair. So, real significant changes. Every one of these cells are haploid cells. So, something to keep in mind when you're comparing meiosis versus mitosis, I like to do it through those drawings, that page that I created for you, where you're doing a comparison of mitosis and meiosis. Where does it occur? So, meiosis versus mitosis, remember these are body cells. These are where you're going to produce your gametes, and that's happening in specialized areas. When is it needed? How many types of offspring cells are produced? How many and what type? And how many rounds of cell division or cytokinesis takes place? So you can take a break and figure that stuff out, and then catch up with us. Okay, so here's that same diagram that was way back in the beginning of the lecture. And so if you want to print something like this out, then you can add some information in there if you feel like it's necessary. Keeping in mind that these are haploid cells. Fertilization takes place. We return back to the diploid state, and then it's mitosis from here on out. Happy little couple with their happy wee screaming baby, although it's not screaming. Okay, so here's the answers to the last frame. I'm going to pass right by that. So, what sexual reproduction does is it leads to variation. And the way that it leads to variation is, one, is that independent assortment. So, how those homologous pairs are going to line up side by side in the center of the cell, called independent assortment. And then random fertilization. So which of those 4 eggs are actually going to be the one that, gets fertilized? And a process called crossing over. So I think that we're going to look at those more specifically, and so we'll start with independent assortment. Sorry if I seem a little confused on my slide presentation. It's because there's a big break for me between when I make the first video and the second video. So, but anyway, that's neither here nor there. So independent assortment. So this concept right here leads to a lot of variation, and this is a lot why what we're going to go through right now is the reason why you don't look exactly like your siblings. So if you have a brother or a sister, no doubt you look a lot like them if you came from the same parents, because you're shuffling the same information, but which of that information you get really creates a lot of variation. And so the processes that create that variation, one is called independent assortment. And so let's go through that concept right now. So it has to do when the chromosomes are lining up during meiosis 1 as homologous pairs. So when they come to the middle of the cell, the metaphase plate, how they come, how chromosome pair number 3 here lines up with dad's on this one side and mom's on the other, they get to line up independent of what this chromosome pair did, independent of that one. So they're just as likely to have dads over here and moms over here. 50-50 chance that moms would have ended up on this side and dads would have ended up on that side. And depending on how they line up there can give you a lot of variation in your offspring. Because if this, say, is our hair color, chromosome number 1 was determining the hair color, let's say that this chromosome number 3 is determining your right-handedness versus left-handedness. So my mom had blonde hair, and she was right-handed. And my dad had black hair, and he was left-handed. And so if the chromosomes line up as they do in this diagram here, when this cell splits down the middle, then some of my gametes are going to code for my mom's blonde hair, but my dad's left-handedness. Where other of my gametes are going to code for my dad's black hair, but my mom's right-handedness. Now, should they switch positions so that mom's is over here and dad's is over here, that's what we're talking about, independent assortment. This pair here gets to do whatever they want. This pair does not control what is going on with any of the other pairs. So if those two reversed and the red one was over here, then when these split down the middle, then I would be producing gametes that had my mom's blonde hair and information for my mom's right-handedness, and then I would have gametes for my dad's black hair and my dad's left-handedness. Something like that. That's exactly what's going on. It's called independent assortment. And if you figure out all of the potentials that we have, so here's an example of it. See, here's my mom's. It's possible that my mom's number 2 would line up underneath my mom's number 1, but there's nothing that says that has to happen. Mom's number 1 could line, or mom's number 2 could line up underneath my dad's number 1. And when the cell splits down the middle, that will give me 2 different cells than these. So if I compare these gamete cells that are produced to those, they're going to be significantly different. And when you calculate that out for all of the chromosomes that we have, since we have 23 pairs of them, you can figure out how many different kind of gametes you could make. It's 2 to the 23rd power. That's over 8 million possibility of gametes that you can possibly make by this process alone, called independent assortment. Okay, and then there's the random fertilization of the egg and the sperm, and you'll get an idea of that from the video. If you watch that video on fertilization, you'll get an idea as to how fertilization is completely random. You have no control over which sperm is going to actually get to enter into the egg. And if it's that sperm versus this one, you can imagine